Dr. Farzana Fisher, Rahiman, University of the Western Cape, South Africa. She would give her presentation on the vitro effect of Galenia africana on malignant melanoma and human keratinocytes. Welcome, Dr. Farzana.
Good morning. I'm just going to share my presentation with you quickly. Okay, good morning, everyone. Thank you for having me. I will be presenting a paper that was published by my lab here in Cape Town, South Africa. And the title is The In Vitro Effect of Galenia africana on Malignant and Non-Malignant Skin Cell Lines. So just to begin with, the skin, as we all know, is constantly exposed to UV radiation from sunlight, which causes cell damage resulting in cancer. And melanoma, as our key speaker has already mentioned, is an aggressive malignant form of cancer with a very high metastatic ability. In fact, it accounts for nearly 73 to 80% of skin cancer-related deaths, with an 18% chance of a 5-year survival rate. Now, there are various stages of melanoma. The early stages are predominantly treated with a combination of surgery and other therapies, including chemotherapy, radiotherapy, and so forth. But as we know, as much as those forms of chemotherapy can be beneficial, it's also associated with some adverse effects. And so it really makes standard treatment very difficult due to the increased majority of melanomas ultimately developing this resistance to both systemic and conventional cancer therapies, eventually requiring the use of a combination of treatment options. And so the administration of such therapies, such as chemotherapy, etc., is usually really restricted to specialized centers that are not accessible or easily accessible in developing countries like South Africa. Therefore, there's a need for a more effective anti-cancer agent that can be well tolerated by patients, that is non-toxic to the healthy cells, and as well that is something that's affordable and be readily available in poor communities. And so at present, we have seen, although there's a continuous development of novel agents for the treatment of melanoma, but there has been also a shift towards using more natural products. And this is predominantly due to the bioactive or the phytochemicals that are present in these plants, which has now been deemed as the most promising and novel strategy to prevent or inhibit or reverse the process of carcinogenesis. And we know that the use of herbal medicine has, over the past decade, increased dramatically, with about 64% of the world's population now using it. And now South Africa is not just a beautiful city, but a country, but we also have a very rich diversity of indigenous medicinal plants, with an estimated 3,000 species being traditionally used. Well, that's what we know of. And over 27 million people in South Africa reportedly use this traditional medicine for healthcare. In the Western Cape in South Africa, we have an indigenous group that is referred to as the Khoisan, or the bush doctors, and they just have a wealth of knowledge about the plants around them and how to use these plants for treating different skin ailments, not just skin ailments, but various other conditions. And so we really just wanted to tap into that knowledge. And in order to assess that first, we conducted a preliminary survey, and those results will be published soon. But just to give us some insight on what are the types of plants that can be used to treat different skin infections. And that's just a snippet of our preliminary survey results. As you can see, there's that's just from one family. There's just a lot more types of plants that can be used. And that specific table there was just relative to fungal infections. But one of the plants, through conversation with these indigenous leaders that came up, was the plant Galenia africana. And this is quite an interesting plant. It's known as kraalbos. It's predominantly found in the Makwa land in the Western Cape, and it's used, as you can see listed there, for various conditions ranging from coughs, wounds, tuberculosis, skin infections, inflammation of the eyes, and so forth. What's very important to note that is the biological phytochemistry of it has been done on this plant, and the compound that was identified was pinosembrin, and pinosembrin is a major flavonoid that was found in the ethanolic extracts of the Galenia africana, and it has been shown to possess some sort of anti-cancer activity. So the aim of the study was to investigate or explore that further. We used 2 cell lines, and that is the malignant human melanoma cell line, which is the A375, and then we use the normal human cell line, which is the keratinocytes or Hackett cells. And our objectives was basically to compare the anti-proliferative or the proliferative effect, because we didn't know what the plant was going to do. Obviously, look at cytotoxicity, because with plants at certain dosages, perhaps at the lower dosage versus the higher dosage, they could show a beneficial or a toxic effect. So it was important for us to assess that concentration range. And of course, to look at the morphological changes within the cells. So this is just a brief overview of the methodology. As I said, we use those 2 types of cell lines. In terms of preparation of the plant, because this plant is commonly available within the Western Cape and used by so many people, it has been commercialized. So we purchased it from a local company. We used a 20% extract, and that was gone through a vacuum drying chamber to dry the ethanolic extract. So there were so it didn't have any other effects on the, or the ethanol didn't have a adverse effect on the cells. In terms of looking at endpoints, we looked at the cell viability, and we used 2 tests for that, 2 in vitro tests, which is the MTT and the trypan blue. The principle of the MTT test, basically it determines the cell viability by measuring the enzymatic reduction of the yellow tetrazolium salt to purple parmesan via cellular mitochondria. So we basically seeded approximately 10,000 cells into little wells, and we exposed them to the plant and then assessed this endpoint using a commercially available kit. The trypan blue assay, which is also a cell viability test, was merely to confirm the results we saw in the MTT assay, and that is but it was based on a different principle, and it's on the principle that live cells possess intact cell membranes that exclude certain dyes, such as trypan blue. And of course, in terms of cytotoxicity, we use the lactate dehydrogenase assay, which is a cytosolic enzyme present in the cells that are released into the cell culture medium when damage is upon the plasma membrane. And finally, the nuclear morphological changes, just to explore if there were any associated changes or that could be related to apoptosis. And we conducted this using the Husting. Okay. In terms of results, statistical analysis was performed using SPSS, and it's important to note that we did replicates to ensure that the experiments were all repeatable, and they were expressed as mean and standard deviations. Okay, and that's the first results of the MTT assay. As you can see, Galenia africana, it showed a significant and dose-dependent, what we refer to as a dose-dependent trend in the A375 cells, which is the melanoma cell lines. And after 48 and 72 hours, 25%, approximately 25% and 60% of the melanoma cells exposed to 500 microgram of the plant extract had experienced cell death. Cells exposed to the 1,000 microgram for 48 and 72 hours showed a reduction of 38% and 74% respectively, and that can be seen in this figure, in figure A. In contrast, the Galenia africana extract showed an upward trend and then a decrease when exposed to the Hackett cell line after 48 hours. And for this time point, a significant increase can be seen. So there was an increase in proliferation, suggesting that perhaps the lower doses, which is 100 to 200 micrograms, could potentially increase cell growth, while the higher concentrations did reduce cell viability. And the trypan blue assay in some way did confirm the MTT results, because again, it showed that dose-dependent, time-dependent trend with a significant decrease at the higher concentrations of 500 and 1,000 micrograms. But it did this for both the cell lines. In terms of the LDH assay, it confirms our previous results where no cytotoxicity can be seen in terms of the lower concentrations. And that was for the 100 and the 200 micrograms for both cell types that was observed. The percentage cytotoxicity in the A37 cells exposed for 72 hours showed a 55% and a 59% significant increase in the 500 and 1,000 microgram dosages, whereas the Hackett cells only showed a significant increase in the highest concentration in terms of cytotoxicity, and that was roughly 63%. And finally, looking at the nuclear morphology using the histone. So we know that apoptosis is a form of cell death that involves vesicle formation, nuclear membrane blibbing, and chromosome condensation. The histone is merely a stain, so these results would have to be confirmed with further tests. This was just observation of those changes that I've just mentioned. And the A375s, I'm not sure whether you can see this clearly, but the A375 cells exposed to the 500 and the 1,000 microgram per mole actually show irregular shaped and brightly stained nuclei, including nuclear condensation and fragmentation. At lower concentrations, the 100 and the 200 microgram mitotically active cells, they had no nuclear changes could be observed. And the Hackett cells, which is in this diagram, exposed to various concentrations of Galenia africana extract showed very little to no effect in terms of a morphology of the nuclei, with only a few apoptotic features that could be seen at the highest concentration, which is 1,000 micrograms per mole. And that was after 48 and 72 hours, so the longer exposure time. Finally, in terms of our discussion, these results are not presented, but the inhibitive concentration percentage, or the IC50 value, represents the concentration of the extract that is required for 50% inhibition of cell viability. And we find that for each cell line, it's notable that the extract was able to reduce cell viability of the A375 cells, which is the melanoma cell line, at a concentration of 572.7 micrograms. Whereas with the Hackett cells, it was a bit higher, which was 711. And that was after longer exposure. So what does this tell us? Well, the results show that the longer exposure of the melanoma cell line to the Galenia africana results in the greater reduction in cell viability, while the longer exposure to the normal skin cells doesn't have much of a cytotoxic effect on the cells. According to our analyses, we find that the higher concentrations as well, the 500 and 1,000 microgram per mole, reduction in cell viability at all exposure times. And we just, and again, that confirmation that it was done in a time and dose dependent manner. In fact, a similar study also reported that the extract reduced cell viability of malignant breast cancer cell line, the MCF7. And so there is that sort of comparison between the two. However, there was just limited information in terms of the skin cells. When comparing the cell viability effects of Galenia africana when cultured with the normal cell lines at the low concentration, we see that there is very little cytotoxic effects, but an increased proliferation of the cell. So this perhaps suggests that the extract is not harmful to the healthy cells at those concentrations. Again, there has been studies that show that it had reduced cell viability of non-malignant fibroblast cells and breast epithelial cells, and did not show sort of or induce a hypersensitivity response or cause any dermal sensitization to the epidermis. Regarding the apoptotic induction, the direct mechanism was not completely understood or highlighted in the study, which is one of the limitations. However, that will be conducted in a follow-up study. But the Hooch staining did prove that the extract induced associate apoptotic features, including nuclear shrinkage, condensation, and fragmentation in the melanoma cell lines. And the extract only displayed apoptotic features in the Hackett cells at the highest concentration. So the results also support the notion that the plant extract could possibly induce apoptosis in melanoma A375 cells, and the research proposes that perhaps cellular apoptosis could be responsible for inhibiting proliferation of the cultured cells, and defects in apoptosis are important in the pathogenesis of melanoma. So this information perhaps tells us that there is a specific mechanism that the plant has of discriminating between cell types by targeting the melanoma cancer cells while not being cytotoxic to the healthy cells at a low concentration, because we did see an effect at the high concentration in terms of cell viability. The plant flavonoids could interfere also with multiple signal transduction pathways that limit proliferation. And if you remember, I'd mentioned that one of the important flavonoids that was isolated from the plant was pinosembrin. And pinosembrin has been found to trigger apoptosis while suppressing autophagy in cancer cells. It also has the potential to inhibit, delay, block, or reverse events associated with carcinogenesis. And so it's worthwhile investigating or exploring this plant further. Finally, in terms of our conclusion, like I said, the plant has demonstrated selective growth inhibitory, including cytotoxic effects on the viability of the cells, and they warrant further studies on the underlying molecular mechanisms of the plant. And we just need to explore this in terms of our future studies and address cell apoptosis further. And with that said, I would like to thank the National Research Foundation and the University of South Africa for their support in this project. Thank you.
Thank you, Dr. Farzana, for the